Good afternoon. I would like to thank the organizer for inviting me to share my data with you. And my lab at UMass is working primarily to develop systems biology approaches to dissect complex regulatory networks of antiviral immunity. And in my lab, we're looking on two aspects of antiviral immunity. One is the innate immune responses and also adaptive immune responses. When innate immune responses develop very quickly within hours, and it's primarily non-specific, and it's the first line of defense, and the adaptive immune responses, T and B cells responses, they're much slower, and the response of immunological memory. And today I would like to focus on the innate immune side of the antiviral response when the innate immunity provided by dendritic cells and macrophages detect. It's not working. Is that right?
Can we get one of these changed, or both?
Okay. Yeah, that might work. Great.
Just make sure you, I think the bottom button turns off. Make sure you
Okay, that works. Thanks. So when we're looking on the innate immune side, macrophages and dendritic cells, they primarily sense presence of virus or vaccine adjuvant, right? And then signals through TLRs to develop a large transcriptional response to drive cytokine production. And the innate immune system instructs adaptive immune response by T cells and B cells, and primarily shape the magnitude and timing of adaptive immune response. The recent years brought a field of systems vaccinology and also systems biology when we start to recognize that complex networks underlying this response, and they control production of cytokines and shape specific timing, duration, and the frequency production of cytokines. And these tiny changes, dynamics, is extremely important to drive appropriate adaptive immune response by T and B cells. And this actually provides a very important avenue for drug development since we can reprogram using different adjuvants, a combination of adjuvants, we can reprogram innate immune response to shape. Also, we can produce different drugs to reprogram specific networks. And today, I would like to introduce you to our systems biology approach. It's a little bit different from what's existing in the field. And I will give you an example of what we discovered recently. It's a novel pathway driven by FOXS3 transcription factor. And I will give 4 aspects of its regulation and transcriptional epigenetic level, and what's the role of FOXS3 and IR7 circuit in the regulation of in antiviral response in the macrophages. Overall, during the last 10 years, or even more, right, we discovered that viral components would interact with a large number of receptors, either member-associated or cytosolic or endosomal, called TLR, NLR, or RLR factors, to drive a large single transactional cascade that would result in activation of specific transcription factors, like NF-κB, IRF, or STAT. And these transcription factors would translocate to the nucleus and activate specific gene targets. It's overall called antiviral program. It's around 1,000 genes going up and 1,000 genes would go down. And despite the progress in this field, we still don't know the many names of transcription factors that drive the response. So most of the research has been focused on NF-κB, IRF, and STAT factors. We don't know much about negative regulators of the response. We don't know much about epigenetic regulators. And that's exactly where my lab came in. We developed an approach that uses a combination of transcriptomics, proteomics, and epigenomics. And when we use this combination of all these 3 omics approaches together, we identify specific circuits. We predict specific regulatory circuits that usually it's 3-5 transcription factors, epigenetic factors that drive a certain cytokine production. And we use mathematical modeling to predict their dynamics and validate these findings in vivo and also in ex vivo models. So just to simplify, we contrast different factors like TLR agonists, like in this case, poly-IC, LPS, and PAM3, to activate different TLR responses. And when we're looking on the transcriptome profile, we identify specific co-regulated genes. Like cluster 1 will be induced, generic response induced by all 3 TLR agonists, and cluster 2, it's antiviral response driven primarily by TRIF pathway. In this panel, we developed a huge data set that including multiple flu infections in different cell types, and we identified specific models that would be activated by certain agonists. And our question was, when we have these gene models, we ask what exactly the regulatory circuits would drive the expression of these models. And we use an algorithm to predict specific transcriptional epigenetic regulators, and I won't go into details. We found that unexpected that FOXA3 transcription factor is a key regulator of IRF7 pathway, and IRF7 is a master regulator of antiviral response in macrophages and dendritic cells. And that's a pretty unexpected finding because FOXA family is primarily known by regulating metabolic responses. And it been never implicated until recently in the regulation of innate immune response. What makes the finding really important that the FOXA family also implicated in the development of immunological memory in T cells. Overall, just to give you background, the FOXA3 it's primarily controlled by AKT pathway. It's inhibited by AKT pathway, and the FOXA control autophagy. That's a positive driver. So it's known as a positive driver. So very, very, very few data suggest it as a negative regulator. And actually, our data show that the FOXA3 in macrophage and dendritic cells function as a negative regulator of antiviral response. Deletion of FOXA3, when we examine the FOXA3 now, macrophage and dendritic cells, we observe upregulation of a large number of antiviral genes in the basal state, and also the response was even further enhanced by poly-IC stimulation. And when we looked what is the gene that's responsible for the phenotype, we actually focused on IRF7. That's the most deregulated gene in the macrophages. And as I mentioned to you, IRF7 is a master regulator of antiviral response. So we show that that FOXA3 binds directly to IRF7 promoter, and the ablation of FOXA3 results in increase of IRF7 under basal conditions. The binding of FOXA3 to IRF7 promoter we observe in two sites, in the proximal to transcription start site and also in the distant. And deletion of this transcription factor binding site resulted in increased transcription of IRF7. So just to summarize the first part, we observed FOXA3 keeps in check IRF7 gene. The question was then what the mechanism underlying FOXA3 mediated repression of IRF7 pathway. And since FOXA3 is a transcription factor, we decided to focus on the epigenetic component of FOXA3 mediated repression. In general, histone acetylation is associated with increased levels of transcription, and histone deacetylation promote repression of gene transcription. And that's why we hypothesized that FOXA would recruit deacetylases to repress IRF7 gene, and that was because of this finding that we observed that FOXA3 deletion resulted in increased levels of histone acetylation at the IRF7 gene, as you see by a red line, in contrast to blue line in wild type. Okay. So we used protein-protein interaction data sets that we developed in our lab to predict partners of FOXA3 in epigenetic, that's proteomic studies, and we predicted 8 potential histone deacetylases that would associate with FOXA3. We validated all of them, and then actually turned out that FOXA3 associates with HDAC3 with NCOR2, that's nuclear repressosome complex, to repress IRF7 pathway. What makes the story really important? It turned out that FOXA3 it's not alone. It's actually more than 200 proteins that co-regulated with FOXA3. And we call it butterfly genes. As you can see, these proteins they're present at the high basal level in macrophages and disease. Upon antiviral response, their level would drop down. You see by blue color, the expression is down, and then they recover back. So it's a very unique profile. And and many of these proteins been already shown to associate with innate immune responses, and with different defects and their deletion associated with different defects of memory T cell formation. I will just go quickly through the story. So we did multiple biochemical studies to validate that FOXA3 interact with NCOR2 repressosome complex, and this complex represses not only IRF7 but a number of antiviral genes. and deletion of, so when we try to delete different components of, like in this case, HDAC inhibitors of this complex, we recapitulate a phenotype of FOXS3 deficiency. So meaning that it's, the results are reproducible, but we can ablate different nodes of this pathway. So what we observed, just to summarize here, is that the FOXS3 would recruit NCOR2 HDAC3 complex to keep in check IRF7 gene and to prevent its autoregulation. And the ablation of FOXS3 would result in uncontrolled IRF7 production, and that would drive antiviral responses under basal conditions. So this approach, we actually use this approach. So the main point of our approach is to predict the important factors and then to validate their significance in vivo. And in this study, we used a model that was developed by Shizuo Akira. And that's, in his model, he showed that VSV, it's a VSV virus infection in lungs would result in alveolar macrophages. So in interferon response in alveolar macrophages. And we use his model to identify the role of interferon in lungs. So as you can see in FOXS3 null mice, we observed, when we introduced intranasal poly-IC, we observed severe damage in lungs. It was associated with neutrophil influx. And the same was by VSV infection, and that was associated with massive neutrophil influx. When we switched, so we decided to compare how this whole circuitry behaves. So we decided to contrast FOXS3 and IRF7 null mice. And as you can see here, that's actually the main point of this paper, that in response to viral infection, in FOXS3 null mice was a transient damage in the lungs, and this damage was resolved by day 5 of infection, whereas in IRF7 null mice, we didn't observe so we observed a lack of inflammatory response, and by day 5, we observed significant lung edema, so the lungs got filled with fluid, and these mice actually died when the FOXS3 null mice recovered. And that's just to show you the two arms of the response. When FOXS3 null deficiency resulted in a decreased viral load. Oops, it also died. That's funny. So as you can see here, the decreased viral load in FOXS3 null mice was associated with increased lung damage, whereas in the IRF7 null mice, the lack of viral control was associated also with lack of inflammatory response. And then to show you that the two arms of the response are extremely important. And when we play with both arms, we can actually tailor the appropriate response, both innate and also we can tailor adaptive immune response in mice. Just that's a summary slide just to show you where we go with this project. We decided to focus on both sides, innate and adaptive side, when one goal is to improve vaccine efficiency by working on vaccine formulations. When measuring the response in different vaccines, we can actually predict what would be the best vaccine formulations to enhance adaptive immune response. And we're also working on the systems immunology side, more looking toward innate immune response to predict how different viral virulence factors would reprogram transcriptional circuit for their benefit. And I would like to thank two groups. My group at the UMass Medical School. That's the project was primarily driven by Aaron Lampana, a senior scientist that works with me more than 10 years, and Amy is a graduate student in my lab. And I would like to thank my ex-postdoc PI, Alan Adir in Seattle Biomed. So he established an Institute for Systems Biology with Leroy Hood, and he primarily introduced the systems biology approaches to and established system vaccinology and systems immunology fields. And the Ilya Shmulevich group also at the Institute for Systems Biology in Seattle that helped with computational analysis that I did present. Okay, thank you.
Any questions?
Yes?
Can I ask if you've ever had a look at just reduced expression of FOXP3? Because I think there's a human polymorphism in the gene which leads to reduced expression, and that gives you decreased susceptibility to malaria but increased likelihood of developing severe autoimmune diseases. I think the explanation given is that's all through TGF-beta. But I wonder if maybe they're wrong.
Right. That's a really good question. So FOXA3 is associated in Tregs. It's right Tregs, FOXP3 pathway.
I think there's a Fox.
Also FOXA3. Yeah. They didn't say pathway. FOXA3 drives FOXP3 expression in Treg, and it actually was shown that specific by human genetic studies. There are specific mutations in the FOXA3 gene that was associated with increased susceptibility to malaria, but at the same time, it's actually different phenotype is Crohn's disease. So it's actually you get either. So that's you tweaking the immune response. So and the same with viral responses. So we just starting to look into it. So this pathway is absolutely new. So it's very few people look into this pathway. We have human genetic studies and very few immunological studies. Yes.
Sorry, I might have missed. Does IRF7 deleted mice, do they show deleted lungs? Do they show more neutrophil influx?
Yes, so they show more neutrophil influx, even though now we do believe that it's more macrophage-driven phenotype. At the beginning, so if you compare our earlier data for neutrophils, you see neutrophils, but we do believe that it's more macrophage influx that drive the damage.